this is important both scientifically because it can potentially allow us to more directly investigate the neural basis of a specific behavior, but it also has, of course, neurotechnology implications because it can potentially allow us to better decode and modulate behaviorally relevant brain states. So why is it that it's important to jointly model neural and behavioral data? Let's think of an example experiment in which you're recording some neural activity, you're measuring some behavior you're interested in, let's say arm movement, and you want to find out the relationship between the two. Now, the problem is that not all of the latent brain states that are reflected in neural activity will relate to the behavior that we're studying. For example, if I'm talking about arm movement, maybe some of these latent states relate to my emotion at the time of movement, or how hungry or how thirsty I was. So what we really want when we want to study the neural basis of a specific behavior is to extract and study those latent dynamics and states that are shared between neural activity and the behavior of interest, essentially those green, behaviorally relevant latent states. But unsupervised dynamical modeling frameworks that are trained on neural activity, unsupervised with respect to behavior, may actually find those red states that, while of course exist in our neural recordings, are not directly behaviorally relevant, and they may actually mask or confound those green states. So with this motivation, back in 2020, we published a paper on a method called preferential subspace identification, or PSID, that can directly extract these green behaviorally relevant states to do so more accurately. So the backbone of PSID algorithm was a linear dynamical system model. It wrote down the neural activity and behavior in terms of a latent state and describes its evolution over time with linear dynamics. Now, the capability that was provided by PSID was that it could dissociate the element within the state that were behaviorally relevant from all of the other elements to learn them more accurately and with much lower state dimensions. So the algorithm is published. I'm not going to go into the detail, but just to give you a little bit of intuition, the intuition is that these green states should summarize everything you know about future behavior in terms of past neural activity. So PSID essentially projects future behavior time series onto past neural time series. We show by doing this, we can extract a shared dynamical subspace between the two and then fit the model parameters within that subspace. Importantly, once you fit the model, then the extraction of these latent states in a test set has nothing to do with behavior. Essentially, you just take neural activity in, you run a very simple, fast, efficient Kalman filter with parameters that are learned with PSID, and then you extract your latent states. So how does this work? We tested PSID on multiple algorithms, including a reach, grasp, and return movement data sets from Bijan Pessaron's lab, where we were measuring 27 joint angles, as well as spiking and LFP activity from over 100 channels. And then we compared the behavior decoding of PSID with a neural dynamical system model that was trained unsupervised with respect to behavior, and then downstream trained the decoder to movement. And what we found was that PSID was more accurate in decoding of movement regardless of the state dimension. And more importantly, it could do this accurate decoding using a much lower dimensional latent state, so in this case, 5 times lower. So what this indicates is that by dissociating the behaviorally relevant dynamic, first, we can learn them more accurately. Second, we can reveal that their dimensionality is actually much lower. And this benefit held even when we compared with nonlinear dynamical system models that were trained unsupervised with respect to behavior, such as sequential variational autoencoders. And this is, again, because of this new capability for dissociation. Okay, so this is great, but another important question that arises when we think about neural computations or dynamics that give rise to behavior is whether those dynamics are intrinsic to a given brain region we're recording from, or they're due to dynamically structured external inputs. And in a lot of cases, we may have at least partial measurements of these inputs. Let's think of a motor task. In this case, for example, you may have task instruction, which target you want the monkey to reach to. That is a sensory input that you're measuring. If you think about neurostimulation, whether optogenetic or electrical, this is another form of input measurement. And finally, maybe you're recording from two brain regions and one is input to the other, so you have some sort of measurement. So the question is, can we, and if so, how can we incorporate measured inputs into our joint neural behavioral dynamical modeling framework? Now, I'm showing you a graphical depiction of the problem formulation. As I mentioned, we can think of this problem as having behaviorally relevant states and other states that give rise to behavior and neural activity. The question now is how the dynamics of these states are generated. There are two possible sources for this. One is intrinsic dynamics that reflect the connectivity within the neuronal population you're recording from. But there's another source, which is dynamically structured in external inputs. So if you look at this in the frequency domain, you can think of intrinsic dynamics as the transfer function for this input-output box. You can think of inputs that come in and they have some frequency spectrum and they convolve and then they give you the neural spectrum. So if you look at the neural spectrum, it has a peak corresponding to the transfer function or intrinsic dynamics, and it has a peak corresponding to the external inputs. So can we dissociate the two to better tease apart the neural computations? How do we do this? All right, so first, obviously, we have to include the input within our model. But the capability, the new capability we need to provide is that we need to dissociate the behaviorally relevant subtype of intrinsic dynamics, both from input dynamics as well as other intrinsic dynamics. And as I'll show you, we need to do so while prioritizing the learning of this behaviorally relevant subtype, because that is critical to its accurate identification. So how do we enable these new capabilities? The basic idea is that we formulate a learning problem that simultaneously involves measured neural, behavioral, and input signals. And then we develop a multi-stage learning algorithm to solve it, in which we can, in earlier stages, prioritize the learning of the behaviorally relevant subtype. Okay. So how does the algorithm work? The derivation, and this is an analytical algorithm, so the derivation is a little bit involved, but I'll give you a little bit of intuition how we do this. So we have three signal sources: neural activity, behavior, and input. We form future and past time series of these inputs. Then in an earlier first stage, what we do is we project future behavior time series into past neural activity and input time series, but then in an oblique manner along the direction of future input. The intuition being that your future behavior is generated according to intrinsic dynamics that operate on the past as well as future inputs. So somehow, if you want to extract the intrinsic dynamics, you have to cancel out the effect of future inputs inside your operations, and that's how we do it. But that won't be sufficient. We'll show that we further need another projection onto the past as well as the future, and then through a singular value decomposition, we can extract these intrinsic subtype of dynamics that are behaviorally relevant. Now, if you tell me, okay, I want the other intrinsic dynamics too, that's no problem. In a second stage, we then project future residual neural signals, again in an oblique manner, along the future inputs onto past signals, and we can get the other states. Once we do this, then we can fit the model parameters. Okay, so how does this algorithm operate? How does it work? First, we wanted to confirm that it's accurate within numerical simulations, artificial data. So we simulated models, and we found that all the model parameters were learned with errors that converged to 0. So that was great. The next question was, how accurate are we in identifying these intrinsic behaviorally relevant dynamics? So to look at this, we quantify dynamics through the eigenvalues of the state transition matrix, because that tells us how your state moves from one time step to the next. And we simulate here a toy example where your system, your model, has two behaviorally relevant states, two behaviorally irrelevant states, and the input has two states. So first, we train a neural dynamical system model, or NEM, that is trained unsupervised with respect to behavior and cannot take into account input. And of course, the modes or the eigenvalues we found were confused by both inputs and other intrinsic dynamics. Then we train PSID, and in this case, we are no longer confused by the intrinsic dynamics that are not behaviorally relevant, but we are confused by inputs. Then we say, okay, let's fit an NDM, but take input into account. And you can do this using various types of approaches, expectation maximization, subspace. There's a lot of ways to do this. So again, you don't get confused by input, but by other intrinsic dynamics. And finally, the only method that can actually correctly identify these behaviorally relevant intrinsic dynamics is this method. So this is a toy example, just to provide some intuition. But we did extensive simulation to actually quantify the error of the method versus other approaches. So here's the eigenvalue error as a function of the training samples. The systems we simulate here have two behaviorally relevant states and 6 total states. And this is how a neural dynamical model that is provided with input and takes input into account, but is unsupervised with respect to behavior operate. You can see that our model with just 2 states is quite accurate. If I fit an INDM with 2 states, it is pretty bad. That's the top line. So I have to go to higher state dimension to be able to find the behaviorally relevant states somewhere within those higher state dimensions. So we fit one with 6, and you can see that it's still much less accurate, and much less accurate at every single number of training samples for every data set. In particular, it needs orders of magnitudes more training samples to get the same accuracy. So you may ask me, why is that? So the problem is, because there is no notion of prioritizing one subtype of dynamics over the others, INDM needs to learn a much more complex model and associate some model capacity to other intrinsic dynamics. And that means that it's much less accurate in the subtype we're interested in, and therefore needs much more training samples. So this shows you the importance of prioritizing the learning of the behaviorally relevant intrinsic dynamics. To show more evidence of this, we then said, okay, let's implement a numerical optimization approach that in a single stage tries to optimize the log likelihood of neural and behavioral data given input. So it has all of the signal sources that our method has access to, and the same issue arises. It was much less sample efficient, and it was much less accurate at a given data set. So this again indicates the importance of this multi-stage learning. So having fully validated our model within artificial simulation, we went ahead and applied it to public data sets. So this is a public data set from the SABIS lab in which monkeys are reaching to random targets within a grid while neural activity is being recorded. So here, behavior is the cursor position and velocity, and input is the task instruction sensory input that we can actually measure, so target position instructions. So we looked at the dynamical eigenvalues, I'm going to call them dynamics from now on, and we find that we find distinct dynamics compared to the alternative methods. And the dynamics we found with this new method were actually more accurate in both behavior prediction and neural prediction, consistent with our simulations, showing that we can better describe the intrinsic dynamics of neural and behavioral data. Now, this was one data set. But the unique capability here is that we can account for differences in input across tasks. So we said, okay, let's go and look at 2 different tasks in 2 completely independent data sets. So to do this, we also looked at a public data set by the Miller lab, in which now the second pink monkey was actually performing a random target reaching movement with a manipulandum. And we said, let's first look at overall intrinsic neural dynamics, which we can extract with INDM, input dynamical modeling. And you can see that the intrinsic dynamics are different between animals. So 2 animals are doing the same task, which is the Sebi's task. 1 is doing the Miller task. So you can see the intrinsic dynamics are distinct. As you would expect, these are doing different things. These animals, they may have different internal or cognitive states while doing this movement. And then we say, okay, so let's look with our method what the behaviorally relevant subtype of intrinsic dynamics looks like. And they were remarkably similar across animals and across tasks, unlike the overall intrinsic dynamics. So this shows you, by taking into account differences in other intrinsic dynamics, as well as differences in terms of input across tasks, we can actually study intrinsic neural dynamics or computation across different tasks and come up with dynamics that we would not otherwise find potentially. So this is a useful tool for studying intrinsic computation across different behavioral contexts. All right. So far, I've been talking about this joint dynamical modeling of neural activity and behavior. But the backbone of everything I talked about so far is a linear dynamical system model, which is a great model because it's very interpretable, very simple, very fast, very useful for neuroscience. It's great. But we know from a lot of beautiful work by this audience that nonlinearity is actually quite important in capturing neural dynamics. For example, in unsupervised modeling, and I've listed some papers, there's many, many more. I apologize if I haven't listed some. So that's number 1. Number 2, if we want to go beyond motor neuroscience, we need to deal with behaviors that are not continuous valued. For example, discrete values such as decision choices in cognitive neuroscience. And unfortunately, this linear dynamical system model cannot extend to any of this. So we cannot use it. To do a nonlinear model that can take into account different behavioral modalities, we thus had to go back to the drawing board and write this relationship between neural activity and behavior in terms of a model with 4 interpretable mappings. A mapping from neural activity to the latest state, the dynamics of that state, and then the mapping back to behavior and to neural activity. Now, within a linear dynamical system model, all of these mappings are linear. But as I mentioned, we wanted to make these mappings nonlinear. But we didn't want to just train some nonlinear model. We wanted this model to afford some interpretability in the sense of where the nonlinearity can be summarized across these 4 interpretable mappings. So we wanted to allow each of these mappings to be made linear or nonlinear, independent of the other mappings, so we could do hypothesis testing. Further, as I mentioned, we wanted to allow for categorical behaviors and behaviors that could be intermittently observed. And finally, because we wanted this to work for naturalistic movements and naturalistic behaviors, we wanted to not have our model require a specific trial structure. So we developed a new learning algorithm that can capture all of these flexibilities. The model architecture in this case is a recurrent neural network with parameters that each correspond to one of these 4 mappings. The input to this RNN is the neural activity, and the output is the prediction of neural activity and behavior. Okay, now how do we make this linear or nonlinear in each parameter independent of the rest? What we can do is we can either make each parameter be a linear matrix multiplication or be a nonlinear mapping in the form of a feed-forward neural network. So that is the model architecture, but the question is, how do I train it with neural behavioral data? In particular, I want to train it in a way that I can dissociate behaviorally relevant dynamics. So we developed a dissociative learning algorithm that consists of 4 optimization steps. The first 2 steps extract the behaviorally relevant state within this nonlinear model, and their mapping to neural activity and behavior. And the last two optimization steps extract the additional states and their mapping to neural activity and behavior. Now, the connection between these optimizations, the cost function, all are kind of involved. They involve depending on different modalities. But all of this is in the preprint that we posted back in 2021, so you can refer to that. But what I'm going to show you is what we can do with this method. All right, so first, we met all of these flexibilities, right? We captured nonlinearities. We can now, in our loss, address intermittent samples by masking. We can change our loss to categorical loss to take into account behaviors that are categoricals. So we are ready, and we can go and apply this to diverse data sets. So we picked 4 independent data sets. The first one was the reach and grasp movement data sets from Bijan Pesseron's lab that I've shown you before. The second one was a saccadic eye movement data set in which monkeys were saccadic to one of 8 possible directions. The fourth and the third one were the public data set that I've already shown you with the Miller and Savi's lab, monkeys reaching to random targets. So this allowed us to look at different brain regions, different behaviors, and we also looked at different neural modalities. We looked at spiking, raw LFP signals, and LFP power bands. So first, we wanted to establish essentially 4 use cases for this tool. The first use case was accurate nonlinear modeling of neural behavioral data. So we went ahead and compared the decoding of behavior between this nonlinear method and the linear PSID method that we know does very good decoding compared to other unsupervised methods. And we saw that the nonlinearity actually improved decoding in all of the modalities, not only spike, but also the LFP modalities. This also allowed us to ask, okay, how much does each modality benefit from the nonlinearity? And we found that the most benefit was actually for the raw LFP modality. And interestingly, after doing this nonlinear modeling, the decoding of raw LFP was better than our traditional LFP power band features that we typically use. So this shows you that this model can automatically extract nonlinear features that are more behavior predictive. And in some cases, these features could even do better decoding than spiking activity. All right, great. So we can extract the behaviorally relevant neural dynamics better. What about other neural dynamics? At the end of the day, this is a neural behavioral model. So we looked at the scatters of decoding of behavior versus neural self-prediction. So models on the top right corner are on the performance frontier. They have both better decoding and good neural prediction. And we found that this new method, compared to alternatives such as unsupervised RNNs that are nonlinear as linear PSID, were on the performance frontier across all of the 4 data sets, thus establishing the first potential use case. The second use case was nonlinear dimensionality reduction. So we then compared this method in terms of decoding with an unsupervised nonlinear RNN at the low dimensional regime. And you can see that performance is higher, meaning that we can summarize behavior information in fewer latent states, thus achieving nonlinear dimensionality reduction. The third use case was the localization of the nonlinearities within our model, which could afford some level of interpretability. In particular, we wanted to see whether not only can we just train a nonlinear model, but in addition, see where the nonlinearities can be summarized across these 4 mappings. So why is it that our method allows us to do this hypothesis testing? The 2 properties it has that allows us to do this. The first property is that we can compare a nonlinear source not only with a linear model, which is not sufficient, but also with other sources of nonlinearity so that we can rule out incorrect nonlinearities. The second property is that we can compare different nonlinearities, not only in terms of decoding of behavior or neural prediction, but in terms of both. Because 2 nonlinearities could be indistinguishable in one measure, here decoding. But when you look at also self-prediction of neural activity, you can distinguish them. So first, of course, we went to artificial data to first validate whether this works. We generated data from models where we knew where the nonlinearity was because we simulated them. And we found that in every case, the model found the correct source. Here I'm showing you a model where the nonlinearity was in the neural drive or K parameters. And you can see we find it because it's on the top right performance frontier corner. This shows you why the first property is important, because had I just compared with a linear model, I might have incorrectly assumed that the nonlinearity is the behavior readout, but that is obviously not the case. We also simulated a model where the neural readout was nonlinear, and again, the model found it correctly by finding it on the performance frontier. This shows you the importance of the second property. These models are indistinguishable in decoding, but we can distinguish them when we look at neural prediction. Similar results held where the dynamics were nonlinear, we found them. When the behavior readout were nonlinear, we found it. So let's go to our data and see what we find. So first, we looked at the reach and grasp moving data set with monkeys, remember, from Bijan's lab. So we found that, interestingly, summarizing the nonlinearities just in the behavior readout from the dynamics was largely sufficient in well-description of neurobehavioral data, meaning it was as good as a nonlinear model with flexible nonlinearity anywhere, and it was better than every other individual nonlinearity. So this is interesting. What about the other data sets? So when we looked at them, we found consistent result, that in all of them, summarizing the nonlinearity here was largely sufficient. So this is an example of how doing this affords some interpretability. Now, I can hypothesize that finding this nonlinearity at the output of these motocortical areas might be consistent, for example, with cognition playing an intervening role between the state and behavior. Then I can expand my recording regions, go record from another region, refit this model, and try to narrow down the source of nonlinearity. So this is another useful tool we can use to afford this manner of interpretability. All right, finally, we wanted to see generalization across different behavioral modalities, including cognitive and discrete modalities. So we looked at task epochs, which are categorical, and again, similar results held with this method being on the performance frontier and doing more accurate decoding and neural prediction. All right. So that establishes our use cases. So one other topic that I want to touch on before ending is our efforts to move beyond correlational validations of dynamical models towards causal validation, by which we would apply a closed-loop perturbation and ask whether our model can predict the causal effect of that perturbation on the neural response and the behavioral response. And that brings me to the topic of closed-loop control, which is another major trust in our lab. So how do we envision such a closed-loop control system for validation? We envision a system that would take the neural population activity, decode from that an underlying state of the brain, use that as feedback within a feedback controller to adjust the perturbation, let's say electrical stimulation, and take the brain to a target state. Now, this model-based system, as I've depicted here, is unrealized to date. And there are multiple challenges we have to overcome to realize this system. The first challenge is that we have to build a dynamical encoding model that tells us, okay, how is this state even represented in brain activity? And that's what I've focused on so far, talking about there's a lot of beautiful work from this audience on this front. But unfortunately, for this causal validation, that is not enough. Because even if you have a perfect encoder and a perfect decoder, you still don't know how to perturb the brain to take it to a target state. So what we need to do additionally is to build dynamical input-output model that would describe the effect of external perturbation on neural population activity, and then plug them into a control strategy. How do we do this? So I'll briefly show you some work on challenge 2 and 3. So first, the input-output modeling wants to build a model of this bug, how perturbation, in this case electrical stimulation, would change multi-regional brain activity. So this was a focus of a recent work we did in collaboration with Bijan Petron's lab to show that we can indeed achieve this modeling and prediction. How did we do this? We again relied on our linear dynamical system backbone and included this time the stimulation input into it. There's a good reason for this. These linear models are amenable to building closed-loop control algorithms, and which is notoriously difficult for nonlinear models. So the question here was, how do we fit this model? Of course, we have to collect some data first, stimulate the brain somehow, record the response, and use the data set. But how do we stimulate to get informative data? So in a 2018 paper, that was a theoretical paper, we showed that we needed novel waveforms. In particular, we needed waveforms that stochastically switched the frequency and amplitude of this biphasic pulse according to a coin flip, because these inputs were wide spectrum in the parameter space, could excite the brain persistently such that they could then give us informative data sets. So we went ahead and applied this waveform, this new novel binary noise modulated waveform in Bijan's animals. We simulated in one region, we recorded across a multi-regional network, fitted our models, and we did see that indeed these models could predict how neural activity changes in response to a time-varying stimulation, thus starting to address the second challenge. Finally, now that we have an input-output dynamical model, of course, we have to plug it into a control strategy. How do we do that? So you have your model. The input-output part of that model would tell you how changing the perturbation or stimulation would change your neural activity. The decoder part of this model would tell you how that change in neural activity would change your behavior. So together, this model tells you the effect of stimulation or perturbation on behavior through neural activity allows you to build a feedback controller. So you can run this feedback control algorithm in real time. You can predict what the behavior will be in response to perturbation. You can look at it. Does it match? If it matches, you've causally validated your model. So that's a step that we are currently actively working on. We are also extending these algorithms to become multiscale, so go beyond a single spatial temporal scale of activity, just spikes or just LFE, but look at them all simultaneously together, essentially multimodal and multiscale dynamical models. We have a body of work on this that I won't go into, but a recent work showed that once we build this multiscale, multimodal dynamical model, we can actually find shared multiscale dynamical modes that were shared between spiking and LFE activity and were dominantly predictive of movement. So you can imagine if there exist these multiscale modes, perhaps we can do better encoding, decoding, and control if you identify them and target them across multiple spatial temporal scales. So that's a step we're working on. Finally, a big future work in our lab is to go towards nonlinear modeling within this closed-loop control framework. As I mentioned to you, building nonlinear control is notoriously difficult. That's why we rely on a linear backbone. But can we come up with nonlinear dynamical model architectures that are amenable to flexible inference and closed-loop control? That's a step that we are currently working on, and very excited about that. All right, finally, all of this focus on basic science application. There's a whole direction on neuroengineering and engineering application that I've so far ignored. Of course, these models are useful for brain-machine interfaces. For example, back in 2018, we showed that we can use these dynamical models to decode mood variations from human intracranial brain activity. So you can imagine that doing so gives you a personalized signal to personalized therapies, for example, in depression, by building a closed-loop stimulation system that regulates abnormal mood states, and there's a lot that can be done there. So with that, I would like to conclude. This is some of our work at the interface of machine learning and control and neuroscience to do both basic science as well as develop neurotechnology. And the most important slide is to thank the students in the lab who do all the work. Parsa Wahidi, who's here, is going to present a poster on the input-driven dynamical system modeling for neural behavioral data tonight. So feel free to go and check the details of that out. And I would like to, of course, thank the funding sources. Thank you again for the invitation. It's an honor and pleasure to be here with you today. Thank you. Okay. Okay, I finished on time, so we have time for questions.
Yeah, we have at least 5 to 6 minutes for questions.
Yeah. Thanks for the talk. It's very inspirational. Did you think at all about the multitask problem? How would you preferentially detect a subspace that is behaviorally relevant for multiple different tasks, not just reaching? Like, are we losing something when we kind of focus on subspace that is specific just to reaching? And if we would have another task for that same animal, would we lose the ability to decode something like that?
Yeah, so that's a great question. So the question is, what is your problem formulation? So the problem I formulated here was that I have some measured behavior, and I want to look at the neural dynamics that are shared or give rise to that measured behavior. So let's say you have multiple tasks and there's multiple behavioral streams that you're interested in. So just plug it into your behavioral measurement signal. So plug it into your Z variable. Then you can look at neural dynamics that are relevant to those behavioral measurements that are an expanded measurement. So this framework is general in the sense that it doesn't even have to be neuroscience data. It could be 2 data streams. We're trying to find and prioritize the learning of shared dynamics between these data streams. You define them. If you have multiple tasks, multiple behavioral measurements, plug them into one data stream, and you can identify those dynamics.
And what is the main kind of information that's included in the irrelevant subspace? Is that just kind of general oscillation kind of orbit of the neural data? What is in there?
Yeah, so that's a fantastic scientific question. So you can then, as you said, we don't give you just the behaviorally relevant part, but also the other intrinsic dynamics. And you can look at their eigenvalues, look at their oscillation, time decay properties through those associated eigenvalues. And what they do depends on the context, right? Depending on, for example, in the motor task, maybe those behaviorally relevant dynamics are encoding time into the task or something similar to that. That is a scientific question that then you can use those extracted latent states and try to study them. So this is a tool that can be used. Thank you.
I probably misunderstood half your talk, so maybe this is a stupid question. But one of the things I found very interesting is that you said that the major nonlinearity you needed was from the neural activity to the behavior, right? And then but you could do quite well if you didn't have any others anywhere sort of within the system. But what about things like an attractor, like attractor dynamics within the system, which many people have suggested might occur in these regions? So is that something that you're suggesting is not happening in these regions, or is it?
No, I'm not suggesting that. So I'm saying within these data sets, what we did observe was having and these are motocortical data sets, so having the nonlinearity in the behavior readout was sufficient. It was sufficient to describe the neural behavioral data. I'm not suggesting attractive dynamics do not exist anywhere in the brain. So in these recordings, they were not important for
causally
description.
relevant for these.
Yeah,
this
for
data
these.
set.
So that's why I say this is a tool for hypothesis testing. So this tells us, okay, it wasn't here. Maybe if I expand my recording and include a cognitive area, perhaps there's some cognition that intervenes between brain state in this motocortical region and behavior. And if I include it, I'll find that attractor. And then you'll find it in the A matrix that would need to become nonlinear. The point is, this is a tool for hypothesis testing. In these particular data sets that are movement related, it was sufficient to have the behavior readout nonlinear. But that by no means, of course, it doesn't mean that it always will be the case. So this gives you a hypothesis testing tool. Now you can go and expand your recording and refit the model and look at it again.
Okay, thanks.
We have time for one more question.
Okay, thanks for your talk. In the case of closed loop simulation, how do you deal with the curse of dimensionality in terms of if you have many channels, the possible number of patterns explodes? And also, how do you deal with the huge brain state changes, suppose that in a patient?
Yeah, that's a fantastic question. So we deal with the curse of dimensionality because all of these dynamical system models are designed to reduce dimensionality. In particular, one point I want to raise is that by modeling jointly the neural activity and behavior, we can get a much lower dimensional representation that's just behaviorally relevant. So what you can do in your control algorithm, you can just target that. Forget all of the other intrinsic states. So you can actually minimize side effects by just targeting that state, leaving the other dynamics that could be very useful to be untouched. So that is exactly one reason we developed this dimensionality reduction techniques, in particular the joint behavioral and neural modeling techniques.
Thank you.
Okay.
Please thank Dr. Shanechi for an amazing talk.
Thank you.
Going to connect this up.
Yeah. Okay.
You want to play it in the
Yeah, I'll play it.
full screen? Thanks. Okay. Let's introduce you and you can talk.
Yeah. While I set up my talk, I set up an informal poll asking people if they think spontaneous movements influence activity in early visual areas. And a majority of them responded yes, with a small minority saying it depends on the species. So let's see if we can answer that question in the next 10 minutes.
While he's setting up, this is the next speaker is going to be Chandra Talluri from the National Eye Institute. As always, we can solve very sophisticated problems in neuroscience, but getting stuff to show up on screen is always a very hard problem. So,
I like to get presenter notes and everything as well. So, I'm not getting the presenter notes here. So it's just mirroring, right?
yeah.
Okay.
Either way, it's okay.
Yeah.
Okay.
Started?
Yeah.
Good morning, everyone. Animals interact with their surroundings. This is true from single-celled organisms like ciliates to the more complicated species like humans. Understanding the interaction between spontaneous movements and behavior to neuronal processing is one of the unsolved puzzles of systems neuroscience. With advancements in technology, this has become a big topic in the neuroscience community recently. One study, for example, showed that even when head fixed, mice exhibit a range of spontaneous body movements, and that these spontaneous movements were correlated with spontaneous neural activity in the visual cortex. Another study showed that the neuronal variability accounted for by spontaneous body movements was widespread across the whole brain, including the visual cortex. And more importantly, the neuronal variability at the level of single trials explained by spontaneous movements was much larger than that compared to task-related covariates. And this pattern also persisted in the visual cortex. The studies in mice showed that spontaneous body movements modulate activity in the mice visual cortex. This is surprising because conventional knowledge suggests that primary early visual areas are mainly sensory in nature, and these studies ask us to revisit our thinking, suggesting that the brain integrates self-generated body movements with the ongoing neural activity, even at the level of sensory areas. So We asked if primate visual processing is also modulated by spontaneous body movements. This question has interesting. This question is interesting for a number of purposes and mainly for visual neuroscientists. To this end, we recorded neural activity from early visual areas in 2 macaque monkeys performing visual tasks. In addition to this standard setup, we also used videography to record face and body movements as the monkey is performing these tasks. Even when head fixed and seated in a primate chair, the monkeys in our tasks moved a lot, like adjusting their posture or scratching their butt. Now that we see that monkeys move in our experiment, we wanted a way to quantify these movements so we can relate them to neuronal activity. To this end, we used singular value decomposition method developed by Stringer and colleagues in one of the earlier works I showed before. Briefly, we performed principal component analysis on the face and body recordings at every moment in time and obtain the time courses for each of these orthogonalized components separately for face and body views. We see that these components capture movement time courses quite reliably. For example, component 2 of the face camera seems to capture the area around the eyes largely, and we see that this time course has wiggles whenever the monkey moves its eyes or blinks. So we found that non-human primates show spontaneous movements during task performance, and we were able to reliably extract these movement time courses for further analysis. Now that we have these time courses, we wanted a way to quantify the contribution of movements on neuronal activity. To this end, we used a multivariate linear encoding model inspired by the modeling approach developed by Musol and colleagues in one of the works in mice I showed before. The advantage with this approach is it allows us to quantify the effect of movements on neural activity at the level of individual trials. Briefly, the model contains a set of covariates modeling different events in a session, such as task covariates, which are under experimental control, face and body movement time courses extracted using singular value decomposition method. In addition to this, we also included basis functions to quantify nonlinear, non-stationarities in neuronal firing rate at longer timescales to decouple their effect from events happening at the level of individual trials. We fit this model to the recorded neural activity and estimated the coefficients using ridge regression. And we found that the model does quite a good job at capturing the mean activity across trials, as shown for 3 example units here. And this pattern is also true for across all the recorded population, with the median explained variance close to 97%. So we know that the model works, but what we are really interested in is to quantify the neuronal variability at the level of individual trials. So we computed the cross-validated variance explained measure at the level of individual trials. And here I'm plotting this measure on the y-axis for units sorted by variance explained on the x-axis. Now, if movements were to do something, if we fit a reduced model by taking the movement components out, we expect to see quite a huge difference between the 2 model fits. And we found something similar. The difference between the 2 curves here quantifies the unique contribution due to movements on the neural activity. And we see that similar to the results in mice, movements do contribute to ongoing neural activity, even at the level of individual trials. But there is a wrinkle to the story. Our sessions contain individual trials, where monkeys were required to maintain fixation during stimulus epochs and were allowed to free view during the intertrial intervals. The difference between these 2 is that during the visual fixation epochs, the retinal input to the monkeys is under experimental control because the monkey is looking at the center of the screen and we know the location of the receptive fields. In the intertrial intervals, the retinal input is not under our control because the monkey was allowed to free view. And here is the catch. The results I show here is for those epochs where the retinal input was not under experimental control. When we computed the same metric for epochs where the retinal input was under experimental control, we see that this effect almost completely disappears. And this is also true quantitatively across the population recorded. Since we had two cameras, one facing at the body and one recording the face movements, we were interested if these two movements also show similar pattern. We see that face movements indeed replicate what we found in what I showed in the previous slide. Body movements, however, do not show any modulation in either epochs. And this pattern is also true across all the recorded areas, where face movements show some unique contribution during the uncontrolled retinal input epochs, and body movements do not show any modulation in either epochs. To put these measures into context, these are the unique contribution due to the other covariates that we used in the model, and as expected, task covariates account for most of the variability in the early visual areas. And this is the difference in scales for those interested. To quickly summarize, we see that only face movements contribute to neural variability, and only during those epochs when retinal input is not under experimental control. This raises an interesting possibility. We know that the face cameras also capture eye movements, and our singular value decomposition metric identifies those components that specifically capture area around the eyes. And we also saw that this modulation was only present for uncontrolled retinal input epochs. So this made us wonder if eye movements also explain the observed modulation due to face movements. These are the results I just showed you for the unique contribution for face movements during the uncontrolled retinal input epochs. Now we took the same model, but we included additional regressors that we got from the eye tracker recordings, which include horizontal and vertical gaze position, eye speed, pupil size, and pupil derivative. When we included these regressors in the model, we see that the unique contribution by face movements reduces substantially during the uncontrolled retinal input epochs. And this is also true across all the recorded areas. What this suggests is that including eye regressors almost explained away the variance captured by spontaneous body movements. Now, revisiting the question from the beginning of my talk, do spontaneous movements drive activity in primate visual cortex? Our data suggests that this is not the case, unlike the results in mice. To summarize, we found that when accounting for retinal input, spontaneous body movements do not modulate activity in macaque early to mid-visual areas. It is tempting to speculate the possible reasons for discrepancy between our results and the results in mice. And one possibility is there are genuine species differences. Most of us know that rodent brains and primate brains differ quite a bit in terms of anatomy as well as their function. Indeed, one recent study found that modulation by locomotion is small in marmosets compared to mice when they performed the same analysis on both species. This work was jointly led by me and my colleague Incheol Kang, with contributions from Adam Lazere, Katrina Quinn, and Nick Kaliss, and is a joint collaborative effort between our lab led by Hendrikje Nienborg, Jake Yates at UC Berkeley, and Dan Butts at University of Maryland College Park. Now, before I take questions, I would like to announce that our lab has open postdoc positions, so if you're interested in learning more about the work or to learn about the lab, please get in touch with me or to Hendrikje, who is here at the conference. Thank you.
We have time for 2 questions.
Hi, so maybe I just missed it. I'm wondering, between the controlled and uncontrolled states, how much of the differences you see are actually uniquely neural, and how much of them are actual changes in the behavior itself? That is, do any, fidgets, face movements, eye movements, all these things, how much do those change between the two conditions?
That's a good question. So these are the movement time courses as a function of time at the level of individual trials. And we do see that quite a lot of movements happen during the intertrial intervals, which are in the open, no, which are outside the shaded area here, right? But we did a control analysis to see if the lack of modulation is not due to lack of movements, where, where we split trials into high movement trials and low movement trials by just looking at the controlled retinal input epochs. And if movements were to do something, the unique contribution due to movements in the high movement trials would be higher when compared to low movement trials. But that is not what we found. In fact, we found the opposite, suggesting that movement time courses probably overfit the data.
Hi, thanks for that talk. I have a question. So you showed that eye movements can explain away the effects of face movements. Is your hypothesis then that if you use the eye movements, say, to compute exactly the image that was on the retina at every point in time, that would explain neural activity in visual cortex?
Yeah, that's a great question, which we actually did. So here I'm looking at the uncontrolled epochs, but now, because we know the position of the receptive field, we can infer the input to the receptive field when it is inside the display screen. And we could not infer the input when it is outside the display screen. And we found that even during these uncontrolled epochs, during those time points where we could infer the input to the receptive field for individual units, we see substantial reduction in the unique contribution due to movements. Does that answer your question?
We can have one more question, and then while Anne sets up. Set up. Yes.
Okay, thank you.
More takers, perfect. Our next speaker is Anne Wu from UCSD.
Yeah, and then that's a problem. I need to. I'm gonna bring my own.
You have your own connector? Yeah. Try that as well. Always finicky, especially Zoe.
Yeah, it's working.
She just used her own connector. It worked better.
Yeah.
I think I got for now. All right, get started.
Okay. Good morning, everyone. I'm from Takaki Komiyama lab in UCSD. First, I'd like to thank the Cosyne committee for organizing this meeting. I will now share my work on the targeted stimulation of the motor cortex. Motor skills are learned throughout life. With repeated practice over time, the movement becomes more consistent, such that the same movement can be executed reliably across trials. To understand the neural mechanism underlying motor learning, we simplify it as a 4-limb level task in the half-fixed mice. Previously, our lab has shown that mice can learn the level press task in 2 weeks, which they will develop a stereotyped movement trajectory that is consistent across trials. In the meantime, a specific and stable activity pattern emerges in the layer 2-3 of the primary motor cortex, or M1. Based on these observations, we hypothesized that this consistent activity pattern of M1 layer 2-3 might be underlying this consistent execution of the learned movement. To directly test this hypothesis, we would need to artificially induce the activity pattern and observe its impact on behavior. Towards this goal, we applied the two-photon optogenetic stimulation, which can stimulate an ensemble of individual neurons as shown here with near single-cell resolution. Combined with the two-photon calcium imaging, we can record the activity of layer 2-3 of M1 during the behavior, identify the movement-related neurons, and stimulate a group of cells of defined activity pattern. In this way, we can deliver a specific input into the M1 network. We train mice to push and hold the lever for 1 second after sound cue. Once mice achieve the expert stage, in 20% of the trials, we will replace the sound cue with a brief optostimulation of 20 M1 neurons. We choose to stimulate the early onset movement-related neurons. These neurons begin their activity before the movement starts, and so it is possible that their activity drives the movement initiation. This video shows the example of a mouse performing the task voluntarily. This is a mouse head fixed and sit under the scope. After the sound cue, the mouse skillfully pushes and holds the lever to receive the reward. In the opto trial, we found that artificial stimulation of 20 M1 neurons can induce a highly similar lever push. At least in these trials, by eye, it is hard to tell the difference between the voluntary movement and the opto-induced movement. So this was surprising to us, and we looked into the movement in individual trials in the following analysis. We used the movement correlation to measure the similarity of the movement to the learned movement. This top panel shows the average level trace of movement in low, middle, and high correlation to the learned movement. Expert mice show highly consistent behavior, and in about 90% of the trials, the voluntarily generated movements are highly similar to the learned movement pattern. In about 10% of the trials, the movements were less correlated to the learned movement pattern. Then we examined the opto-induced movement. We found that opto-induced movements are more variable. The opto-stimulation reliably drives the learned movement in about half of the trials, while in the other half of the trials, the opto-stimulation generates less correlated movements. This variability is interesting because it indicates that the behavior response to the same optogenetic stimulation is variable, even though we are trying to stimulate the same 20 neurons in the same way in each trial. So then we examine the potential underlying causes of this variability of the opto-induced behavior. We first consider the possibility that our stimulation is somehow not reliable. However, when we examine the population activity of the target cells, we found that these cells' activity are reliably and equally driven in high correlation trials compared with low correlation trials. Next, we examine the activity of the other non-target movement-related neurons. We saw a marked difference between the high correlation and low correlation trials. So the downstream neurons are presumably activated by the activity of the target cells activity. They show a long-lasting and stronger activity in the high correlation trials compared with the low correlation trials. To visualize the population activity, we used the PCA to find the 3 PCs of the voluntary trials. So the target cells were excluded from this analysis. We then project both the voluntary trial activity and the opto trial activity into the same PC space, and we found that the opto-induced population activity resembles the natural trajectory. So this indicates that stimulation of the 20 M1 neurons is sufficient to generate a naturalistic population activity trajectory in the downstream neurons within M1. When we separated the individual trials into different groups based on how similar the induced movement is to the learned movement, we found that the activity trajectory is more similar to the voluntary neural trajectory when the induced movements were similar to the learned movement, while the low correlation trial trajectory is less similar to the voluntary trial trajectory. So we quantify the distance between the trajectory and the movement correlation of the opto trials. We found there is a significant correlation between the neural distance and the movement correlation. That is to say, the opto-induced population activity trajectory can predict the induced movement kinematics. So why is there such a variability in the response of population activity in the same opto-stimulation? We noticed that the difference of the high and low correlation trials in the neural trajectory is apparent before the opto-stimulation, as shown here by those open circles that indicate 500 milliseconds before the opto-stimulation. We examined the relationship between the pre-stimulation neural state and the subsequent movement. We found that the pre-stimulation neural state also predicts the kinematics of the movement induced 500 milliseconds later. Furthermore, we found that this correlation also exists in the voluntary trials, that the pre-movement neural trajectory distance can predict the subsequent movement correlation. This result indicates that the preparatory state of the M1 population has a profound impact on the subsequent movement, whether it is induced artificially or voluntarily. The observation that the stimulation of 20 cells is sufficient to drive a learned movement is striking. So it is possible that the mice are so primed in this situation that they make movements so easily that any random inputs can generate the same effect. So we asked whether the opto-stimulation induced movement is input specific. In this setting, we instead stimulated 20 late onset movement neurons. Their activity lacks the movement, so they should not be driving the movement. We found that stimulating these late onset cells does not drive the behavior effectively. This is the level trace of an example session with the opto-generated level trace and the voluntary level trace in the early onset target stimulation session and a late onset target stimulation. We found that stimulating the late onset target generates 20% of trials in total. It generates movements in 20% of the trials in total, and the movement generated with stimulating the late onset targets are not as correlated to the natural movements as the early onset target stimulations. To summarize, we found that stimulating 20 M1 early onset neurons induces learned movement. We also found that the variability of the induced population activity is related to the variability of the induced movements. Furthermore, we found that the pre-stimulation population activity state predicts the induced movement. With the finding, we proposed a model that to generate the learned movement, the M1 needs to start with the correct neural state. From this state, when the network receives the correct input, the learned active pattern is generated, which will drive the learned movement. If the network starts at the incorrect neural state, or if it receives the incorrect input, the M1 wants to generate the learned neural trajectory and the learned behavior. To finish up, I would like to thank my advisor Takaki for his support and lots of input into this project. And I would like to thank the Komiyama Lab, our funding, and the Brookoscope technical team. And thanks for your attention. With that, I'd like to take questions and comments.
Hi, thanks for a great talk. You showed that variability, that pre-motion state predicts both the spontaneous and both the natural and the opto motion. Did you try to cross predict, in a sense, use the pre-state relationship in one to see whether you can infer something about the other?
The templates were excluded from this prediction, so that's a cross-validation. Is that what you're asking?
What I mean that the cartoon you drew at the end says that for both, there is a pre-state and the input, in a sense, can either be your optogenetic one or the natural one. But presumably it's before you give the stimulation, the system is at the same state, whether those, or could be. That's the question, basically, whether if you only analyze, let's say, the relationship between pre-stimulus to the result of optogenetics, whether the division of different pre-stimulus states is informative on what happens in the natural motion.
Oh, so the pre-state, let me go to the slides here. The pre-state neural trajectory distance was measured based on the different trials, pre-stimulation states with the distance between the opto trial and the template movement. The templates are the highly correlated voluntary trials. So basically, we are comparing the state between the voluntary trials, pre-stimulation activity with the voluntary trials activity. Is that?
We have time for one more question, and then. Yeah.
So that was great, thank you. I'm curious about the number of neurons that you stimulated. So you showed things work very nicely with 20. Did you play with the number that you stimulated? And if you did, were the responses graded or all or nothing?
Yes, so we played from 20 cells, 10 cells, 5 cells. Yeah, it degraded when you go to 5 cells.
And thank you.
Let's thank Anbu for her talk. It was amazing.
Can I use that one?
Do you want to use this one?
I guess if it's working.
Works pretty well. Yeah.
There's nothing. Not going. What happened? You maybe, oh, okay, it's fine. There we go.
Our next speaker is Jonathan Green from Harvard University.
Thank you. I'm a postdoc with Chris Harvey and Michael Greenberg. I'm going to focus this talk on our studies on this one specific cell type in the cerebral cortex. and our study is on its role in spatial cognition. But I just wanted to give first a very brief, broader perspective on what we're trying to do, which is that we're trying to understand the functions of the cerebral cortex from the point of view of the cell types that make it up. And it's a particularly exciting time to be doing this because over the past decade or so, through recent advances in single-cell sequencing, we now have a very detailed catalog and comprehensive catalog of the different cell types that make up this structure. And with this catalog, we now have the main question is, what are the functions of these cell types and how do they work together to give rise to cognition? And as I mentioned, I'll focus on this one specific cell type. It's a subtype of somatostatin neurons. Somatostatin neurons are one of 5 major classes of inhibitory neurons in the cerebral cortex. And we were able to study this one cell type using this new tool. It's a cell type specific virus. And briefly what we did is we screened different enhancers or gene regulatory elements for their ability to drive expression in specific cell types. And we found this one, SST44. 44 just refers to the number in the screen that drives expression in the specific subtype of somatostatin neurons. And I'll refer to these neurons just empirically as SST44 neurons. And we wanted to get a first pass understanding of what role this cell type might play, and particularly within the context of spatial cognition. And the task that we used to look at their activity and to better understand this was a simple T-maze task, where based on the cue, the mouse would have to turn left or right at the intersection. So if it was a black cue, the mouse would have to turn left, and if it was a white cue, the mouse would have to turn right to receive a reward. So it's a simple association. The mice are performing this task in virtual reality, so they're head fixed and they're running on a ball. This allows us to image their activity as well as perturb the virtual environment, which you'll find is we make use of that later on. We're imaging this area called posterior parietal cortex. It's an area that we know is required for the mice to perform this task, as well as many other decision-making tasks, both in mice and in monkeys. And the first experiment, just to get a first picture of what these cells might be doing, the first experiment we did was just to look if their activity was different in any way from the other cells in the circuit. So if we just look at the non-SST cells first, here I'm plotting the activity of each cell as one row, and these vertical dashed lines indicate the start of each trial in the T-maze. What you can see is that different cells, which has been reported in the literature before, different cells in the population activate at different points along the trajectory. And as a population, they're very heterogeneous and tile that entire trajectory. Now, if we compare that to these SST44 neurons, just by eye, you can see that they look very different. So instead of this very heterogeneous population activity, they instead activate in these synchronous bursts of activity. And this was not only different compared to the non-SST neurons, but also compared to other SST neurons. They were not labeled by this enhancer. So it appears to be specific to this, really to this subtype of somatostatin neurons. And this is quantified on the right here, simply based on the activity between, the correlation in the activity between individual neurons. Now, we've done a bunch of work to try to understand what is the circuit basis of this correlated activity, these synchronous bursts. I won't go into the details of these experiments. I'll just summarize them in this one slide, which is that we did in vivo 2-photon optogenetics, as well as ex vivo patch clamp electrophysiology, to say that these cells actually excite each other. Even though they're inhibitory neurons, they excite each other through gap junctions between them. And we think that that at least helps to explain why they have these synchronous bursts of activity. But the main point that I wanted to get across today was we see these bursts of activity, but what are they being driven by? Is the mouse doing anything special when these cells are firing? So to give you a sense of what this looks like, I'll show you a few example trials. In the first example, I'll show you a trial where there's low activity. It'll give you a sense of the baseline activity of these cells. On the left is going to be the mouse's view of the maze, and on the right is the average activity of these cells over time. So you can see here is the mouse. It's running in virtual reality. It's running towards the end of the T maze. Now the cue here is white, so it has to turn into the right arm in order to receive a reward. What you can see here is it goes all the way to the end. It receives a reward, and then the screen goes black, and that's the end of the trial. And you can see there's very little activity in these cells throughout the entire trial. And what that tells us right off the bat is that these cells are not just responding to the mouse starting to run forward on the ball, the mouse turning at the end of the maze, the mouse receiving the reward, or different positions along the maze, or just the general visual stimulus of the maze. Now here, I'll show another trial where there's high activity, and if you just look at what the difference in the behavior is between this trial and the last trial, it'll give you an intuitive sense for what is driving the activity in these cells. So again, the mouse is running towards the end of the maze here. It's the same cue, so it has to turn to the right arm again. So it goes into the correct arm, but right here it stops just short of the reward zone, starts turning away, and here it corrects on the second trial and turns back. And on the second attempt, it triggers the reward. And you can see where there's this big activity burst, specifically at the end there where the mouse deviated from the trajectory and corrected to then get to the reward. And so what we did was to look more systematically now at trials where the mouse deviated from its normal trajectory here in terms of its heading. And if we look at those events, you can see there's this time-logged burst of activity in these SST44 neurons, as you might expect from that movie. And if we compare that to trials where the mouse smoothly entered the reward zone, you can see in comparison there's much less activity. Now, what you notice from the movie that the mouse is not just making an error, but it's also correcting for that error. And you can see that's in the average turning behavior of the mouse here, where it's turning in the opposite direction as the error or the deviation. So, for example, if it deviated to the right, it'll turn back to the left to correct for that deviation. And so we asked whether these cells are really responding to that deviation, or are they responding to the mouse's correction to that deviation. So we simply just took these large deviation or error trials and split them into trials where the mouse corrected strongly or weakly for that deviation. And it turns out that if the mouse did not correct strongly, there's actually very little activity in these cells. And it's only when the mouse corrects strongly that we see this very large burst of activity. So what that's saying is that these cells are not responding to the deviation itself, but rather to the mouse's correction to that deviation or to that error. Now you might wonder, is this just a turning response? And we can compare the activity of corrective turns, which is what I just showed, to non-corrective turns, so under conditions where it's not correcting for a deviation. And you can see again that that activity is really enriched for and specific to these corrective turns. So these types of experiments and analyses pointed us to this hypothesis that these cells are activating during these error corrections. And the experiment that we did to further test that hypothesis was, because we're in virtual reality, we can rotate the mouse and cause an error, and then the mouse would have to correct for that and then see what the activity in the cells is in response to that. So here's an example trial of what that looks like. It's the same layout, except on the bottom right here, I'm going to plot the perturbation that we're going to add to the mouse. So initially, the mouse is running unperturbed towards the end of the maze, as before. But right here, we're going to rotate the mouse towards the right. Now that perturbation is off, and the mouse is correcting for that on its own back towards up the end of the maze. And you can see this burst of activity in these cells as the mouse is correcting for now an experimentally induced deviation from its normal trajectory. So we can quantify that over trials. We can see, again, there's this large burst of activity when the mouse is correcting for this experimentally induced deviation. And again, we see very little activity if the mouse is not correcting strongly for that deviation. So it's consistent again with this idea that these cells are responding to the mouse's correction to the deviation or error, but not to the error itself. And just to zoom back out to the different cell types, if we plot this activity across the other SST cells and the rest of the circuit, we see that this activity is really enriched in the specific cell type. And so, and we've also done a lot of control analyses and more experiments to say that this activity really is, at least as far as we can tell, specific to these error corrections. So I've mentioned some analyses that say that this activity is not due to just turning in general. So if we look at turning that's not part of a correction, we don't see that activity. We have done open loop playback experiments that argue against these cells responding just to the visual flow that would be present during these corrections. We've also omitted the reward on a subset of trials, and we don't see a difference in activity whether the mouse was rewarded or not. And we've also just simply looked at whether the overall activity of this population is correlated to the rest of the circuit, and we see that overall it's not correlated, and which would argue against a role in just normalization, so just controlling the excitation in the circuit. So in short, or in summary, these experiments are pointing to this hypothesis, or this working hypothesis, that these cells are responding in some way during these error corrections. Now, to give you a sense of, how are we thinking what function this might serve, one obvious idea is that these cells are just driving the course correction, and so they're serving a role in task performance and keeping the mouse on track. And the second possibility is that they're serving a role in a longer time scale in learning. So one possibility is that they're acting as a learning signal that is teaching other cells in the circuit, this is a correct action and this is an incorrect action, and adjust your weights or your activity accordingly to, over time, favor the correct action. And we think that, isolating this very specific signal in this specific molecular cell type that we have repeated access to with this tool will allow us to better understand this really fundamental signal that can be involved in either of these processes. And I'll just end sort of on the broader scope. Yeah, it's done. We're not just interested in this one cell type, but in all these other cell types and how they interact to contribute to this type of spatial cognition. I'll quickly thank my two mentors, Chris Harvey and Mike Greenberg, who have been super supportive throughout this whole project. Carissa, Thomas, and Jonathan have been, really important driving these experiments forward. Jen and Lisa were involved in the slice electrophysiology experiments for showing that these cells are gap junction coupled. And I'll also mention Sinisa Hrvatin. He now has his own lab, but he was the one who initially discovered the SST44 enhancer. So that's it. Thank you very much.
We have time for 2 questions because I'm keeping the audience between coffee. So 2 questions.
Very interesting results. I was curious to know how specific these error corrections were. For example, in terms of the spatial location that you're applying these perturbations, or the direction of rotation. In the simulation that you showed, I think both times you were doing a clockwise type of perturbation. I was curious to know if, have you tried counterclockwise, for example, and whether the same neuron carries information about where the perturbation was applied or the direction of perturbation was applied. I was curious to know.
Yeah, it's a great question. So I just showed, the rightward perturbation. We did both rightward and leftward, and you see activity in both cases. In terms of position, you can see activity at any point along the trial. So in the first example, for example, I showed it was right at the end in the arms of the T. The second example, it was more in the stem. In natural sort of error corrections, they tend to be at the very end because that's where they need to make this turn. So just the natural distribution will tend to be towards the end, but it can occur at any point along the maze. And I think one question was about if individual neurons might care about left or right error corrections. We've looked at this. It seems like most of them will activate to some degree in both. There's sort of a variability there, so some will activate more to one versus the other, but we don't see clear, clustering of, one will activate towards the right ones and one will activate towards the left ones.
Thank you.
Thank you for the talk, and I just had a very little follow-up for the previous question. So whether you see the amplitude of the signal tracks the amount of error they have been corrected.
Yes, so we've looked at the relation between the activity and the magnitude of the error as well as the magnitude of the correction, and it seems to scale with both of them.
Thank you.
Yeah.
One more question. I'm hearing a strong hypothesis about what would happen if you inactivated these cells. What if you were able to stimulate them? Do you think you would get a sort of aberrant error correction when there was no error made?
Yeah, it's a great question. I mean, right now we're trying the inhibition experiment. But for the excitation, it would depend on if it's driving the correction. You might expect, it sort of corrects when it shouldn't be or something. Yeah. For the learning hypothesis, I think it would be a little bit more nuanced, and you'd have to, better understand when you're stimulating in relation to what. But yeah.
Awesome. I'm excited to see the inhibition results when you get them.
Yeah. Thank you.
Thank you all the speakers for an amazing session. Thank you. We have I think, just a 10-minute coffee break. Yeah, exactly. And we should be back here at 10:45 so we can get our next session started. Thank you, everyone.
seats. Okay, welcome to the second session of the morning. We have 3 talks, the first of which is from Tatyana Engel at Princeton and is entitled Identifying Mechanisms of Cognitive Computations from Spikes.
Thank you, Ashok, and thank you for organizer for this amazing opportunity to share our work at Cosyne. It's the most receptive audience. I feel very excited. What we are interested in is to understand how cognitive functions can emerge from neural activity. And we all know these cognitive functions, they actually a product of interactions between excitatory and inhibitory neurons in the brain wired in neural circuits. Traditionally, scientists build handcrafted neural circuit models, which instantiate specific hypotheses how excitatory and inhibitory interactions can give rise to intelligent behavior. Here is an example of one of the classical models. It's a model for 2 alternative decision making, where 2 excitatory populations, E1 and E2, compete with each other through a common pool of inhibitory neurons. As a result of this competition, one of the populations will elevate the firing rate, indicating the choice. The power of this type of circuit model is that we have complete understanding of how connectivity of the circuit gives rise to its dynamics to produce the behavioral outcome. In particular, we can understand these models analytically, for example, in the phase plane. In the phase plane of this dynamical system, there are 2 attractors, one representing one of the choices, and the flow field will drive neural activity towards one of those attractors to create a binary outcome. It's very powerful because we can understand how this attractor structure will depend on the connectivity of the circuit, and in particular predict results of perturbations to this connectivity. For example, we can make predictions of how decision-making behavior will change when we change the excitation and inhibition balance in the circuit. So we have tools to test these precise predictions in perturbation experiments. However, such handcrafted neural circuit models come short in capturing the complexity and heterogeneity of neural responses which we observe in the brain. Neural recordings in the cortex typically show that responses of neurons are very diverse. They vary both in time. The same neuron responds to a combination of multiple task variables, and each task variable triggers response in entire population of neurons. These properties of the neural code distributed mixed selectivity is very hard to reconcile with simple functional clustering of neural responses in handcrafted neural circuit models. It seems the complexity and heterogeneity of cortical responses it seems incompatible with simple mechanisms which we pose in our neural circuit models. To understand how it can arise in a neural network, a new approach emerged where we could train a recurrent neural network to perform the behavioral task of interest. And interestingly enough, in this type of networks, we will also observe neural responses which show distributed mixed selectivity. However, these networks also have complex connectivity, and the connection between connectivity and dynamics is not so obvious in these models. As a result, it's hard to understand what mechanism is generating the behavioral outcome. There is a common idea that RNNs trained through backpropagation discover qualitatively different task solutions than simple solutions in interpretable neural circuit models wired by hand. So how can we understand neural computation in such systems with distributed and mixed neural responses? A common approach emerged to understand these computations in a neural population state space where each axis corresponds to activity of one neuron in a population. Each point in this neural space will represent a pattern of activity across the population. And then coordinated changes of this activity will trace out a low-dimensional neural manifold which will implement the computations necessary to solve the task. Neural manifolds were very successful over the past couple of years to compress this daunting complexity and heterogeneity of single neural responses to reveal very interpretable low-dimensional pictures which can be related to task behavior. This success led to the idea that maybe neural manifolds are the necessary and sufficient building blocks to understand cognitions. At its extremes, this view even suggests that maybe understanding of cognition on the level of connectivity and circuits is not necessary or even impossible. So in my talk today, I will try to bridge between these two perspectives, the neural manifold and neural circuit perspectives on cognitive functions, by showing some of the recent work from my lab. The first part of the talk is a statistical approach, which was developed by Mikhail Genkin, a postdoc in my lab, to identify single trial dynamics and try to link it to a mechanistic perspective on neural computation. And the second part of the talk, which was done by another postdoc in my lab, Chris Langdon, is trying to actually develop a framework. How can we think about circuit mechanisms in systems with distributed mixed selectivity? So we will start with one of the most basic cognitive functions, perceptual decision making. When we make a perceptual decision, we need to convert a sensory stimulus into a categorical choice. For example, in this experiment performed by our collaborators, Chandrasekaran and Krishna Shenoy, a monkey needs to select red or green target depending on the dominant color in the checkerboard stimulus composed of red and green squares. Such binary decision computation can be described by a one-dimensional decision variable, which reflects the evidence accumulated towards one or another choice option. Although the accumulated evidence is the key component in most behavioral models of decision-making, we still don't understand the mechanism which lead to the decision formation in the brain. There are several factors which make it challenging to identify mechanisms of decision computation. First, decision unfolds with a unique time course on single trials. We can register the final choice which the animal makes when the decision variable reaches one or another decision boundary, but we cannot reconstruct these decision trajectories by simply averaging over multiple trials. Second, it is unclear what equations should describe the dynamics of decision variable. Behavioral models often assume a simple drift diffusion model, in which there is a deterministic drift, which represents the strength of sensory evidence towards one particular choice option, and a stochastic component modeled as noise, which captures the trial-to-trial variability in the decision formation. This model indeed can provide a good fit to behavioral data such as speed and accuracy of decisions, and moreover, it can capture some features of neural activity. In particular, if we average these decision trajectories over multiple trials, in trial average responses, we will see ramping activity, and the slope of this ramp will correlate with the difficulty of the sensory stimulus. If we examine single neurons in multiple brain areas, we can indeed find neurons which also show this characteristic ramping activity in trial average responses, as shown here in these example neurons from monkey dorsal premotor cortex. However, it was also pointed out that trial average responses not necessarily capture what happens in individual trials. For example, ramping can arise from drift diffusion process in firing rates, or it can arise from a process where neural state lingers in undecided state for variable amount of time and then transitions to one of the two decision states almost instantaneously on individual trials. And if the times of these transitions vary from trial to trial, it can also give appearance of ramping activity in trial average responses. And moreover, if we look across many neurons, this canonical ramping activity will be rather a rare exception. And across a population, we will see heterogeneity of time courses, and it's still unclear how to reconcile these heterogeneous dynamics with a simple decision computation. So we thought that we can maybe capture the decision computation as dynamics evolving on a low-dimensional manifold. But to be able to reconstruct these dynamics from experimental data, we need to develop an approach which can satisfy several criteria. First of all, we need to have an approach which can learn dynamics on single trials. Second of all, we need to discover equations which control the dynamics directly from data without making specific parametric assumptions such as ramping or stepping activity. And finally, our framework should capture heterogeneous relationship between the common population state and responses of individual neurons. You can think about these relationships as tuning curves of individual neurons to the latent population state, very similar to how neurons in sensory systems have tuning curves to sensory stimuli. We went on to develop an approach which would be able to perform all these functions. So I will give you a brief idea of how our approach works. Think about neural trajectories in some latent population state space. These trajectories could be very complex, but if we know the equations of motion which generated these dynamics, they become very interpretable. In these equations of motion, you can think about deterministic force, F, as defining some potential landscape in which dynamics evolve, and the noise adds variability to these dynamics on single trials. This model is intrinsically interpretable because if we have description in form of this general dynamical system, we have all the tools from dynamical systems theory to understand how these dynamics will behave. So now imagine we can morph this landscape of dynamical forces continuously in any arbitrary shape. And in this way, we would be able to explore a continuous space of hypotheses about latent population dynamics. So our goal will be to learn the shape of these dynamical forces directly from data in a space of continuous functions. Our framework, as I already mentioned, assumes that there is a latent population state in which dynamics are described by these dynamical system equations with noise. And then to account for heterogeneity of single neural responses, we implement unique firing rate functions or tuning functions, one for each recorded neuron, which map common population state to firing rate of individual neurons. So you can think again about those functions as tuning functions to the position on the common manifold. Both the dynamical forces and the tuning functions are known to us, and our goal will be to learn these functions directly from neural response data. Because we work with spiking data, we assume that given the instantaneous firing rate, spikes are generated as a Poisson process. Let me illustrate you how this flexible inference framework can learn different kinds of dynamics. Here is a synthetic data, is a population of only 16 neurons, which were generated by following a common population state shown in the gray line. In this example, the population state alternates between two states stochastically and lingers for some variable amount of time in each state. Although it's a very simple dynamics where dynamics are switching between two states, it's not very obvious to see this transition in spiking responses due to Poisson variability. Our model can accurately reconstruct these discrete transitions by only analyzing spiking data. But also, if the ground truth dynamic would have a different shape, for example, a smooth continuous Gaussian process, our model would be able to identify these qualitatively different kinds of dynamic. How does it do it? Because these different types of dynamics correspond to different shape of the potential landscape in our dynamical system. These continuous transitions correspond to dynamics which have two attractor wells, and noise drives stochastic transitions between these two states, creating appearance of jumping or stepping dynamics, while continuous fluctuations correspond to variation of activity around the single attractor state. Our framework learns the shape of this potential landscape from spike data. Here is an example situation on the ground truth data, which is shown here in gray lines. We generate data from dynamical system, which has two attractors. And there are three example neurons in this population, which have different shapes of their tuning functions to the latent population state. The ground truth is shown in gray, and the colored line shows the current estimate of our model about all these components of the framework as we use gradient descent algorithm to optimize all of these components simultaneously. You see that starting with unspecific guess, our model is able to smoothly morph into and find the solution which indeed generated the data in this case. Within this very general framework, the previously proposed ramping and stepping model of decision-making are just as special cases of the potential landscape. In particular, the ramping or drift diffusion dynamics correspond to potential landscape which has a constant slope, with a slope pointing to the boundary corresponding to the correct response. And noise can occasionally drive the dynamics to the opposite boundary to generate an error. Here, the blue line shows the initial state distribution, is the distribution of the latent state at the trial start, which is also unknown, which we also infer from data in non-parametric way. The stepping model, on the other hand, corresponds to dynamical landscape in which there are two barriers, and the particle starts in the attractor in the middle, and to reach either of the decision boundary, it needs to jump over the potential barrier, creating the appearance of stepping dynamics. So our model can reconstruct either of these possibilities when the ground truth data would be generated from these types of models. But of course, we were curious to find out what type of dynamics do we observe in the brain. It is not necessary that the brain dynamics should conform to one of these ad hoc hypotheses, so we were very interested to apply it to actual neural recordings data. We analyzed the dataset collected by Chandrasekaran. He recorded from monkey dorsal premotor cortex while monkey performed the decision-making task, which I described to you. And as I already mentioned, if you look at individual neurons in this dataset, they show a lot of heterogeneity in their trial average firing rates. First, we analyzed single neurons in this dataset using our framework. So we fitted dynamics of every individual neuron one at a time. And then we analyzed populations of neurons which were recorded simultaneously. And in this case, we had access to small populations, anywhere between 4 and about 23 neurons recorded simultaneously. So I will first show you results of fitting individual neural responses. Here's an example neuron in PMD, which is choice selective. You can see the separation by choice in its trial average firing rate. It's almost a canonical neuron. When we fitted dynamics of this neuron with our framework, we found a potential landscape which was not consistent with either ramping or stepping hypothesis. Instead, this landscape had a smooth, almost linear slope towards the boundary corresponding to the correct choice. And it had a potential barrier on the way to make an incorrect choice, on the way to make an error. To remind you again, the blue curve shows the initial state distribution. So you see that we start on the smooth side of the potential, leading to the correct choice, and errors will require to jump over a potential landscape. When we fitted other neurons in our dataset, about 150 neurons in total, what we found that even neurons which had more heterogeneous responses with time-varying firing rates, also this neuron shows the signature of the very same dynamics of the potential with a single barrier. And all the heterogeneity was absorbed in diverse shapes of tuning functions of these neurons to these simple one-dimensional dynamics. We then apply this network to neural population recordings, and here is an example of one such population. It's only a population of 8 neurons, and in this population you also see diversity of time courses and preference of neurons for left versus right choice. And when we fitted neural population responses, we fit a common dynamical system, but unique firing rate functions for every individual neuron. The result we found by fitting neural population responses was very similar. The dynamics again showed the signature of potential landscape with a single barrier, while all the heterogeneity was absorbed in different shapes of tuning functions. So at this point, we thought, what does the shape of potential landscape tells us about the mechanism of decision computation in PMD? And here we can refer back to the neural circuit models, which are very well understood. Coming back to this example of a decision model where we have 2 populations competing for the choice, we can look in what is the structure of dynamics of this system in the phase plane, where the 2 axes correspond to activities of 2 competing populations. In the phase space, we have 2 attractors representing alternative choices, and these attractors are separated by a saddle point, and the separatrix going across the saddle point divides the flow field. So that on correct trials, and the orange dot shows here the initial state, will smoothly follow the flow field to land in the attractor corresponding to the correct choice. But on incorrect trials, the noise has to drive dynamics of the system across the separatrix to reach the attractor corresponding to incorrect choice. When the noise drives the system against the separatrix, it goes against the flow field, which is the same as going up the potential landscape, jumping over the hill. To make this argument a little bit more precise, we reduce dynamics of this system to a one-dimensional dynamics by projecting it on the unstable manifold of the saddle. It's a line connecting the two attractors in the phase space. And in this one-dimensional representation, we can compute the effective potential landscape controlling dynamics of the system. And indeed, we find a landscape which has one smooth slope going towards correct choice and a barrier on the way to incorrect choice. We also generated synthetic responses from this model and applied our framework to learn the shape of the potential landscape from synthetic data. And we found that in attractor network, indeed, we have the same shape of potential landscape as we found in single neurons and populations in PMD. So it turns out that, to summarize this first part, we developed a flexible and intrinsically interpretable framework for discovering neural population dynamics from spikes on single trials. And we found that single neurons and populations in the PMD during decision-making show the same kind of dynamics despite heterogeneity in their trial average firing rates. And these dynamics were not consistent with previously proposed ad hoc ramping or stepping hypothesis, but instead revealed an attractor network mechanism. So at this point, I would like to move to the second part of my presentation, which tries to link these heterogeneous neural responses more directly to the connectivity structure in the circuit, because a pure statistical approach cannot give us this connection to the connectivity between connectivity, dynamics, and behavior. Here we decided to focus on slightly more complicated decision-making task. It is a context-dependent decision-making task in which there is more than just a single feature of the stimulus, not only red or green color, but also the stimulus is, for example, moving dots, which can move to the left or to the right with varying coherency. And the choice can be guided either by color or motion feature of the stimulus, depending on the context. So as you can see, the context cue can change how the same stimulus should be mapped to responses. For example, in the motion context, the current stimulus shown on the slide would point to the right because the dots move to the right. But in a color context, the very same stimulus should indicate left choice because it corresponds to green target and green is a dominant color in the stimulus. This task is interesting because unlike simple decision-making, there are multiple task variables which all need to interact to produce the correct behavioral response to every sensory stimulus. And as I already mentioned, previous work showed that if we record from neural responses, for example, in prefrontal cortex, during such tasks, we will see a great diversity on the single neuron level, so it's not easy to guess what is the underlying circuit mechanism. To understand heterogeneity of neural responses, a common approach is a dimensionality reduction when we start with a high-dimensional neural activity in neural state space, and we seek a low-dimensional projection of this activity on a set of axes which will represent patterns of activity correlated with each of the task variables. You can think about this projection matrix as a matrix in which columns correspond to individual task variables, to directions in the neural state space, while rows corresponds to, or they show you how each task variable is influencing activity of every individual neuron. So the mixing here happens because neuron can be influenced by multiple task variables as indicated by every row. So usually these approaches try to demix task variables to find representations of variables which are orthogonal in orthogonal dimensions and don't interact with each other. If you think about it, it's a very different concept from what would circuit models suggest. In the circuit models, nodes representing different task variables should interact and influence each other through recurrent connectivity to perform the computation necessary to transform inputs to outputs. And therefore, it was unclear whether this dimensionality reduction method captured the causal interactions in the circuit, which actually causally drive behavior. We developed a dimensionality reduction approach, which we called a latent circuit inference, which compensate for this absence of interactions. Within our framework, we also seek for low-dimensional projections of neural activity. But within this low-dimensional latent space, we would allow the representations of task variables interact and influence each other as nodes in a low-dimensional latent circuit. So here is our model. It also has an embedding queue, which maps activity of each node in the latent circuit to a high-dimensional neural activity pattern in a state space. But within this latent space, we constrain the nodes to follow a latent circuit dynamics. You can think about it as a small neural network which receives task-relevant inputs, and it's also required to generate task outputs. So latent circuit is a low-dimensional model which solves the behavioral task, and matrix Q maps activity of this low-dimensional circuit onto the high-dimensional network. We can understand the mechanism by which the latent circuit solves the task by studying the recurrent connectivity of the latent circuit. Because indeed this connectivity can be sketched out as a very interpretable circuit diagram, which will show how different task variables interact through excitation and inhibition. But this mapping between low-dimensional circuit and high-dimensional network also give us a set of tools to actually go and test the existence of such circuit mechanisms in a high-dimensional system. In particular, because our approach starts by mapping the trajectories of low-dimensional system to a high-dimensional system with a matrix Q, it means that every node in latent circuit corresponds to a distributed activity pattern in a high-dimensional system, which is one column of this embedding matrix. We can differentiate the mapping of the trajectories, which is assumption of our model, to find the mapping of the corresponding flow fields of high-dimensional and low-dimensional system. And from there, taking the argument few steps further, we can derive relationship between the recurrent connectivity of the low-dimensional and high-dimensional system. This relationship between connectivity is very useful because it allows us to translate the strengths of individual connections from low-dimensional to high-dimensional system. In particular, each connection in the latent circuit corresponds to a high-dimensional distributed connectivity pattern in a large network given by the outer product of two columns of the embedding matrix. Therefore, if we can understand the mechanism by which the small circuit operates, we can design perturbations to test this mechanism, low-dimensional mechanism, but then we can translate these perturbations of activity and connectivity onto the high-dimensional system to test for the existence of this mechanism in high-dimensional system directly. We applied this framework to activity generated by recurrent neural networks trained to perform the context-dependent decision-making task. Here is just an example network which we trained to perform this task. And you can see that the network learned to perform the task correctly by looking in its psychometric functions. You can see that on color context trials, the network makes choices according to the color coherence of the stimulus, and it is insensitive to the motion coherence because it is required to ignore the motion feature by task design. After training, connectivity of such recurrent neural network looks quite complex. So just by looking into this high-dimensional connectivity, it is not obvious what connectivity structure gives rise to this task behavior. So what we did, we took responses generated by this RNN trained to perform context-dependent decision-making task, and we fitted these responses by the latent circuit model. This is what we would get as a result of the fit. We infer the connectivity structure of the latent circuit, both recurrent connectivity inputs and outputs, And we simultaneously infer the embedding matrix mapping the latent circuit dynamics onto the high-dimensional system. By looking in this low-dimensional connectivity, we can easily understand the mechanism operating in the low-dimensional latent circuit. In particular, you see this checkerboard pattern of connections going from sensory nodes to output nodes, which implement the alternative stimulus response mappings. And you see inhibitory connections going from context nodes to sensory nodes representing irrelevant information, which will shut down the irrelevant sensory response mapping in each context. It is a little bit easier to understand this mechanism if I just sketch it out as a circuit diagram. So one more time, if you look in this checkerboard pattern of connections, this is a connection which go from motion right and color right sensory nodes to the output nodes representing choice, and the same for the motion and color nodes representing evidence for the left choice. They map to the left output choice node. And then inhibitory connections will shut down one of those mappings. In particular, in color context, inhibitory connections will shut down the sensory nodes representing motion, and therefore these nodes will not drive the output, and responses will be made purely on the color information. This relatively simple circuit mechanism, which we found in RNN, was hypothesized previously multiple times in several publications in work interested to understand the circuit mechanism of context-dependent decision-making. It seems that the simple suppression of irrelevant responses mediated by inhibitory connections could be a plausible mechanism for how RNN solves this task. At this point, you can argue, well, maybe that's the only way how low-dimensional model can solve the task. And that was the only way for a low-dimensional model to feed the high-dimensional neural responses, but high-dimensional RNN implements completely different task solution. Luckily, we have all our theoretical tools to go and validate the existence of this inhibitory mechanism in the high-dimensional network directly. First of all, we can use the relationship between connectivity of the high-dimensional and low-dimensional signal. So the predicted relationship is that if we conjugate the high-dimensional connectivity with the embedding matrix, we should find a low-dimensional structure which should look very similar to what we see in the recurrent connectivity of the latent circuit. And that was indeed the case. After conjugation with the embedding matrix, we find a low-dimensional structure inside the connectivity of large RNN, which correlates very tightly with the inferred latent circuit connectivity. Note that this relationship is not trivial because when we inferred the latent circuit model, we didn't use the knowledge of the RNN connectivity. We only fitted the latent circuit model to neural response data generated by this network. Second, we can test existence of this mechanism in dynamics of the RNN. The latent circuit also provides us a dimensionality reduction tool because we can project high-dimensional activity onto directions corresponding to the nodes in the latent circuit. In the same spirit to other dimensionality tools, we can see that when we project activity on the context or choice directions, which correlate with activity of context and choice nodes in the latent circuit, trajectories separate according to this task variable. That's not surprising. But more interestingly, when we project neural activity onto sensory directions in the latent circuit, for example, direction representing color information, we see that on trials, when color is irrelevant in motion context, shown here in solid line, the activity is suppressed. So what we see in these projections of high-dimensional activity, that representations of irrelevant stimuli are suppressed. And we see a similar picture for motion representations. On color context trials, motion information is gated out in the activity of this network. This suppression is consistent with the existence of inhibitory connections, which we found in the latent circuit connectivity. But the ultimate test of this mechanism is, of course, perturbations. Because latent circuit model is an interpretable low-dimensional model of a task, we can design specific perturbations for which we can predict behavioral outcomes. For example, if we weaken the inhibitory connection from motion context node to sensory nodes representing color, we would predict that this perturbation should reduce the efficacy of the motion context mechanism and make the network sensitive to the irrelevant color information. Indeed, we can take our small latent circuit and perform this perturbation, and as you can see, the psychometric function is changing. In particular, if you look in this part of the psychometric function, under perturbation, it starts to rotate, indicating that the network is becoming sensitive to irrelevant color information on motion context trials. Our mapping between high-dimensional and low-dimensional connectivity allows us to translate these particular perturbations on a high-dimensional network. It corresponds to a distributed activity pattern in a high-dimensional network, and what we can do now, we can perturb connectivity of the high-dimensional network directly. So now we don't make use of the latent circuit model anymore. We just do perturbation to the RNN connectivity, and we predict that we should see the same behavioral outcome as predicted by the latent circuit model. And indeed, it is the case. We can design all different kinds of such type of perturbations by perturbing different kind of latent circuit connections to verify very precisely the existence of this mechanism in high-dimensional system. To conclude this part, we find that circuit mechanisms, which are very similar to classical handcrafted neural circuit models, can exist in distributed high-dimensional systems, but in these systems, each element of this circuit corresponds to a high-dimensional activity pattern on a neural manifold, and this activity pattern can interact and causally influence each other through a low-dimensional connectivity structure. We found that RNNs trained to perform context-dependent decision-making tasks use interpretable mechanism, which is based on suppression of irrelevant stimulus-response pathways. And the latent circuit approach gives us a tool to actually go and causally verify the existence of such mechanism by direct perturbations of network activity and connectivity, bringing the same power which we had from handcrafted neural circuit models to the domain of very distributed high-dimensional networks. So I would like to leave you with a thought that maybe manifolds are not the end goal of what we should study, and trying to link the manifold structure in neural activity to the underlying connectivity structure can provide us with more powerful tools which can generate experimentally testable predictions to verify our understanding of how the system functions. And some of those ideas we summarized in the review article which should appear shortly together with Chris and Mikhail. So that's all. I would like to thank everyone in my lab, but of course, in particular, Mikhail Genkin and Chris Langdon, whose work I presented today. I would also like to thank our collaborator, Chand and Krishna, who not only shared their data, but they also shared all their ideas with us. I would like to thank all our funding sources, and a particular thank you to Cold Spring Harbor Laboratory, which was our home for the past 5 years, and where all of this work was done. Thank you.
Thanks, Tatyana. Also, thanks for being extremely on time. So let's take some questions.
So thank you. That was really elegant work. I'm curious whether in principle you could use your approach to at least infer the sort of the potential landscape from non-simultaneously recorded data, if you made some assumptions about trial similarity. And then, because then you sort of can use all the cells that we've recorded, let's say, over a year, and then project, once you have the matrix, project for individual trials. Will that still work if the landscape is constructed from non-simultaneous data?
For the first part of my talk, we cannot use cells which were not recorded simultaneously. We can fit individual neurons, but the whole concept which we try to implement, that dynamics has a unique time course on every individual trials. And neural responses correlate with these dynamics. And I didn't show it, but these dynamics also correlate very tightly with animal behavior, for example, with animal reaction time. So combining across different recording sessions would defeat the purpose of using single trial activity. For the second approach which I presented, we can apply it to trial average responses, because here we don't try to precisely estimate the equations controlling dynamics on individual trials, but just try to capture the structure of the manifold which generate trial average responses.
Thank you for a great talk. I'm sort of wondering, what are your thoughts on incorporating these frameworks also in cases where you have learning and plasticity?
That's a very interesting question, of course. We didn't do it. Presumably learning would change. For example, thinking about potential landscape approach, you could also wonder, like, is it always the same landscape which describes neural dynamics? And the answer is, of course, not, because even if you change stimulus difficulty, you will affect the speed and direction of decisions. So the way we modeled this, for example, but what we noticed is that the tuning functions to the neural manifold are very stable across different stimulus difficulties. So we fit a model in which tuning functions are shared across task conditions, but potential landscape will be specific for each stimulus difficulty, which can capture differences in dynamics. So what I could imagine with learning is that not only potential will change, but also the shape of the manifold potentially can change as a result of changing connectivity. So I could imagine that maybe in that case, the tuning functions will not be stable.
Hi, thanks for the great talk. I have a question about the first part. So you have there a very expressive and flexible model, right? So that those curves, the tuning curves and the dynamics of the latent dynamics are very, I guess, non-parametric and flexible. So I'm wondering if overfitting is a concern at all, and given realistic amounts of data, number of neurons and trials, etc. So what are your thoughts on that?
That's an excellent question, and I didn't say anything about it, but we wrote a separate paper just on the issue of model selection when you work with very flexible models. Indeed, overfitting happens, and it happens in all kinds of flexible models, not just in our framework. It happens the same way in neural network models. So here you see an example. It's a typical behavior when you keep fitting this framework to a finite sample of data. We get close to the ground truth solution, but then we start to develop spurious features which were not present in the ground truth dynamic, which is a signature of overfitting. But even more interestingly, maybe, you cannot catch this overfitting by cross-validation. And it's related to some of the work in machine learning literature that over-parameterize or very flexible models. Even if they overfit to zero error on training data, they can predict new data very well. And that's the case for our model too. So if you try to use any of the conventional model selection approaches, such as, for example, looking for the minimum of cross-validation curves, it cannot solve the overfitting problem. For that reason, we had to design our own approach, which is based on evaluating complexity of the models which we discover along the way, and then comparing models learned on different samples of data, and only keep the features of these models which agree. It's a complex problem. Maybe I don't give it enough consideration in this quick sweep through the slides, but you can check out the paper. It's an entire paper on overfitting problem.
Okay. So did I get it right that early stopping is a component of the solution?
We use early stopping as criterion, but the question is where to stop. And if you try to choose where to stop using just by cross-validation metric, you will be wrong very often. So for that reason, we had to devise a conceptually different model selection approach, which doesn't look in predictions of the model, but instead directly compared model features learned on different samples of the data.
Thanks a lot. Let's take one more question while Pavel sets up. I don't know which of you was there first. I think it was you.
Thank you for the great talk, Dr. Engel. You said that your model captures response time and other behaviors at the level of individual trials. But as we know, response times also depend on other factors like alertness of the animal that can vary across like multiple trials. Is there a provision in the model to capture those long timescale states and make any predictions on those?
Not in this work. It's not something we modeled. So all trials of the same condition we just modeled them as sampled from the same stationary distribution. So we didn't at this point model any trends in, for example, reaction times, such as slowing down after errors, etc. But it's something, of course, of interest with which we should be looking in the future.
Thank you.
Thank you.
All right, let's thank Tatyana again.
It's not connected yet. Oh.
Yep, good. Okay, so we're going to stick with the Engel lab. The next talk is from Pavel Tolmachev.
I want to thank everyone to come into this session. I'm super excited about sharing our research. So let's jump straight into this. So animals and humans in particular have an ability to flexibly extract behaviorally relevant information from the stimulus. So let me give a concrete example of what I mean. So what is the font color of the following text? So posing the question this way, the answer is red. But if the question would have been, what is the color implied in the text? The answer would have been different. So thus, given the same sensory stimulus, but different context, we pay attention to either one feature or another. So how could this flexibility be implemented in the brain circuits? So one particular task studying cognitive flexibility is context-dependent decision-making. So the sensory stimulus, moving colored dots. Depending on the context, one has to base their choice either on color or motion information. In the motion context trials, if the majority of the dots are moving to the right, make a saccade to the right, and vice versa. Similarly, in a color context trials, red color is paired up with the right choice, green with the left. Thus, this task requires flexibly pay attention only to the relevant stimulus. So the task structure is as follows. The context is presented first, the stimulus comes afterwards, and then the animal has to make a choice. So some of the earliest model of cognitive flexibility were proposing a neural circuit mechanism in which the irrelevant information is suppressed. So the simplest model realizing this mechanism is as follows. So the circuit is supplied with a context information and sensory information. There are two context nodes, context motion and context color. The context nodes inhibit the irrelevant sensory information. So within each sensory modality, motion or color, there are two nodes integrating the evidence, causing the right and left choice separately. For instance, color right and color left. So in the motion context node inhibits the color sensory nodes since this information is irrelevant. On the other hand, in a color context node is activated, inhibiting the motion sensory nodes. So an alternative mechanism has emerged from studying the context-dependent decision-making from dynamical system theory perspective. So the dynamical system theory perspective employed studying large continuous time recurrent neural networks as a model of prefrontal cortex dynamics. So studying the trained RNNs from this new perspective revealed that within each context, they possess line attractors, so green and blue, which are aligned with the choice axis. Here I show 100-dimensional trajectories projected on 3D subspace, spanned by choice, context, and combined sensory axis. The neural trajectories, each corresponding to a different trial, first separate along the context axis and then along the choice axis. So this dynamical perspective gives rise to the selection vector mechanism for performing context-dependent computations. So let's zoom in on a random point on the line attractor within a color context. Consider a linearized dynamics around the fixed point by looking at the Jacobian of this system. From the Jacobian of the RNN around this point, one can compute a selection vector, such that if the perturbation of this network nodes aligns with the selection vector, it would drive the system along the line attractor. Here, the color axis has a non-zero projection on the selection vector. Thus, the perturbation along this color axis, which is orange arrow here, drives the system along the line attractor. As soon as the sensory input is switched off, following the vector field, the trajectory falls back to the line attractor. It converges back to a different point, resulting in evidence integration. Finally, the selection vector reorients depending on the supplied context. In color context, so green line attractor, it is aligned with color axis. In motion context, it is aligned with the motion axis. Now we have two perspectives and two mechanisms. Are these mechanisms truly different? So first we show that the small, handcrafted circuit with inbuilt suppression has the same dynamical picture as the trained RNNs. So it also relies on the selection vector mechanism. And we go the other way, from the dynamic system perspective, back to a small circuit. So now let's get started with the first part. So we handcrafted a small circuit so that it solves the task. So notice some patterns in the connectivity matrix. So first, the output-related checkerboard pattern and the suppression pattern from context nodes. So we performed the same dynamical system analysis of this circuit as we did on the RNNs and revealed the familiar line attractor structure in a small circuit. So that's very similar to what we see in the RNNs. So what selection vector looks like in a small circuit? Here, look at the motion context trial, which starts from some point on a line attractor. The magenta selection vector S has a strong overlap with motion axis. Thus, given the driving the system along the motion axis causes an evidence integration, which is orange trajectory here. If we take a look at the weights of this 8-dimensional selection vector, we notice that they are heavily concentrated on behaviorally relevant sensory nodes and the output nodes as well. So why is that? We can see why from the circuit perspective. The easiest way to drive the system along the line attractor is to directly manipulate the output nodes, so the concentration on the output nodes is not surprising. But the concentration of the selection vector on behaviorally relevant nodes could be explained by the combination of two facts. So the inhibition of irrelevant color nodes below the threshold, that they can not affect the output, and the existence of the structural excitatory connectivity from relevant sensory nodes to the output nodes. Okay. And the same picture goes in a color context. But the elements of the selection vector are concentrated on the color and the output nodes. So notice the rotation of the selection vector across the context. So the dynamical perspective alone cannot demonstrate why this selection vector reorients. On the other hand, the circuit perspective with the suppression mechanism provides a mechanistic explanation behind this realignment. Let's formalize this link further. So the irrelevant nodes do not influence any other nodes at all because they are deeply suppressed. So we looked at the effective connectivity during this suppression. And because of the suppression, the effective connectivity changes, zeroing out the connections coming from the irrelevant sensory nodes. The selection vector is defined by effective connectivity, and thus the inbuilt suppression mechanism causes the changes in effective connectivity, which in turn influences the selection vector orientation. So, but what about the large networks? Do they also rely on this suppression mechanism? And we get to the second part, from dynamical perspective to the neural circuit perspective. So we start with RNN, trained to perform context-dependent decision-making. So the recurrent connectivity matrix, denoted by capital W recurrent, is difficult to interpret. And then we apply a latent circuit inference procedure developed in our lab. Well, thank you, Chris. And okay, the main idea behind the latent circuit inference is, let's say the RNN has 100 units, and each trial produces 100-dimensional neural trajectory y of t. On the other hand, I have an 8-dimensional circuit model with yet undefined connectivity here. So the circuit produces an 8-dimensional trajectory x of t. And then we embed this circuit trajectory x of t into this 100-dimensional space with an embedding matrix Q. So fitting the traces of the large recurrent neural network by circuit responses, we obtain small recurrent connectivity. So this is the first parameter we get, and the matrix Q, embedding matrix Q. Here's some fancy picture. The columns of the matrix Q serve as representations of the latent nodes in the small circuit. I refer to these representations as small Qs. So thus, the activity of each node in this latent small circuit correspond to the activity along the whole Q direction in the RNN. So after running this latent circuit inference, we first analyze the small recurrent connectivity, W recurrent, and we clearly see a suppression mechanism pattern between the latent nodes. Using the obtained task variable representations, so this Q directions, the columns of the matrix Q, from the latent circuit inference, we show that the selection vector of the large RNNs are linked to the structural connectivity of the latent nodes. We take a middle point on the line attractor relevant to the motion context and compute a selection vector. We directly compare the selection vectors of the inferred latent circuit and the 100-dimensional selection vector of the RNN. So projected with the matrix Q to bring it down to the 8 dimensions. So the both selection vectors, so magenta linked to the RNN and the blue to the latent circuit, so they are compatible with one another, indicating agreement between the inferred latent circuit and the RNN. And secondly, they are heavily concentrated on the behaviorally relevant sensory nodes. So these are the same insights we got from the studying the small circuit. So the same analysis within a color context gives similar results. Here, selection vector elements are concentrated on color nodes. Now, does this reorientation of the selection vector, depending on the context in the large RNN, also occurs because of the suppression mechanism? So we compared the projected effective connectivity of the RNN with that of the inferred circuit, and we observed a marked attenuation of the effect of the latent nodes representing their relevant information on the output. In this case, the color encoding directions do not influence the output directions. And the same picture is present in a color context. Thus, in a large RNNs, irrelevant sensory directions has limited ability to affect the output directions. The selection vectors are directly computed from the effective connectivity. The context-dependent changes in the effective connectivity are caused by distributedly implemented suppression mechanism, And hence, the latent suppression mechanism gives rise to the context-dependent changes in the selection vector. So the takeaways. A circuit with inbuilt suppression exhibits selection vector mechanism. It is not an emergent property of large systems. Selection vector arises from the latent excite-free connections. We provide a showcase how the latent connectivity structure gives rise to the selection vector. And then rotation of the selection vector is caused by the suppression of the relevant activity via latent inhibitory connection. So the suppression mechanism in the latent circuit causes the changes in the effective connectivity in the large networks, which then causes reorientation of this selection vector. So thus, our research implies that the latent suppression mechanism may be behind the ability of the large neural networks, be it biological or artificial, to perform context-dependent computations. And this suppression mechanism is consistent with the selection vector mechanism. So finally, I want to give a special thanks to all my lab mates and the source of funding, NIH Brain Initiative, and thank you for listening.
Okay, thanks. One or two quick questions. Go ahead.
Hey, here. I was just wondering, so when, I know this is evidence accumulation, but if I were to look at the sensory areas, I would typically expect the context to come into a different layer than the, let's say, sensory input. And this would basically, I think that this would give you different input dynamics. So I'm just wondering, you're training a recurrent neural network with inherently, it doesn't need to be linear, but the inputs of context and the input of sensory stimulus are somehow similar. They're additive, so to speak. Whereas if you have a context-dependent variable coming into layer 1 and you were computing in layer 2, 3, which I assume is what you're modeling. So yeah, how does this change the, would you expect some sort of multiplicative effect or some sort of more gating explicit effect to change how much you can map the vector, the suppression vector, or?
Yes, selection vector. Not sure I entirely understand the question, but I guess it's the, so you're saying that the context actually comes from the layers before the actual network? Yeah, yeah, I guess, so even if the context comes before, it still has to be distributed via local circuits. So the context, I think there is a valid assumption that we have a context representation within this a local circuitry.
I guess it's not exactly what I meant, but
yeah,
okay, yeah, sorry, I cannot make it clear. Another, if there's nobody else,
yeah, go ahead while she sets up.
It was not, so in the previous talk, the basic underlying model was a point attractor somehow, or at least it looked like that on the poll. Like here you're using a line attractor. What,
yes, so,
how do you differentiate those two?
So this is actually a collection of slow points. So there are still only a handful of true fixed points. But in between, there's very extremely, the norm of the right-hand side is extremely small, but not exactly zero. Oh. So that's what we call line attractor.
Oh, fair.
All right, we have to move on, but thanks.
Another keyboard voice.
By this.
This. Okay.
Yeah.
Nice.
Okay, the final talk is from Camille Roland from NYU.
Hi, everyone. Thanks for the opportunity to be here. My name is Camille Rudang, and I'm going to be presenting work I've done with my advisor, Christina Savin, on the neural and behavioral signatures of inference over time, otherwise known as dynamic inference. So consider a tiger trying to catch some lunch, and it sees a deer. Pinpointing its exact location isn't easy. It's hard to see where it is. And this deer probably doesn't want to be lunch, so it's going to do its best to run away and avoid the tiger. This is a really common, yet computationally very complicated scenario. Both the predator and the prey have to infer, using unreliable information, the location of the other, which is itself changing and intrinsically noisy. Being able to do so quickly and accurately is absolutely critical for survival, and despite how complicated this problem is, both are able to solve it amazingly well. Although in this case, the deer does it a bit better because it runs away. So what dynamic inference does is provide a mathematical framework for how these really complicated and time-dependent computations are carried out. Specifically, it describes how you can use noisy observations to represent beliefs about behaviorally relevant states, for example, the location of the deer. The agent can then use these beliefs to guide actions, for example, by making predictions for where the deer will go next. New evidence coming in may or may not match these predictions, so these internal estimates have to constantly be updated. And if you think about it, the same computational goal is applied to a really wide variety of problems. After all, the state of the world is never fixed. So perception and action have to be able to account for changes in the world over time. And our work tries to understand how dynamic inference might be carried out in neural circuits. To put this more concretely, what underlies the ability to do dynamic inference is first the agent's internal model for the dynamics of the latent state, which we'll call Z, that relates the state at one time to the state at a later time, and captures how noisy this process is. You can think of this as the prior, which answers the question, how do I expect the deer to move? We also need a way of understanding how sensory observations, X, are generated from these latents, as well as an understanding of how reliable these observations are. This answers the question, where do I see the deer? We can put these two pieces of information together to compute a posterior belief over the latent, given all the information we've seen so far, which is, where do I expect the deer to move, given where I've seen it? The approach that we take to model how this posterior is computed in the brain falls under the category of inference algorithms known as particle filters. Under this approach, distributions are represented by particles, which are samples from a probability density. Regions of state space that have higher probability have more particles associated with them, and factors increase with uncertainty, and that noise correlations increase to more strongly reflect stimulus correlations. Our model also makes predictions for population dynamics, but at the moment we don't have access to data to test those. In a second set of experiments, we looked at manipulations of prior expectations of speed. Specifically, we considered two prior conditions, one fast and one slow, and we probed the network with stimuli from an intermediate speed, which revealed the effects of the prior. We also manipulated sensory reliability, which corresponds experimentally to manipulating the contrast of the target. So behaviorally, our model predicts that the ability to track a target presented at the probe speed depends on both the prior and the contrast. So here I'm plotting I speed for all four prior and contrast conditions, and you can see that if the model has a high speed prior, it tends to overshoot the target speed, and that the size of this overshoot depends on the contrast. Okay. This is expected for Bayesian inference when the reliability of your observations decreases, you rely more on your prior. We can summarize this as the percent difference of I speed between the two prior conditions for high and low contrast, and we see that I speed tends to be more different when the contrast is low. And this is consistent with reports in primates from experiments by Darlington, Beck, and Lisberger. Neurally, we find that the firing rates for neurons tuned to the stimulus are more strongly modulated by prior expectations at low contrast. And if we plot a histogram of the firing rates for all the neurons across the different stimulus conditions, we find that this effect holds across the entire population, which agrees with results from the same set of experiments. So somewhat surprisingly, manipulating observation noise had a really small effect on the Fano factor distributions in this case. Understanding why requires looking at the equation for particle dynamics in more detail. The effects of observation noise come in through the gain and the innovation. In the simplest case, the innovation takes this form, where the optimal gain is equivalent to the Kalman-Busi gain. The input varies on a scale of sigma, but the gain is inversely proportional to sigma, meaning that as the noise gets larger, the relative contributions of the observations decrease more and therefore have a smaller effect on neural activity. So this suggests that there could be a systematic difference in the effects of these different sources of noise. Moreover, there's interesting effects in the Fano factor dynamics. So here I'm plotting the Fano factors over time for the high contrast case. After stimulus onset, you see a transient increase in Fano factor followed by the expected crunching of stimulus variability. These transients are actually quite common in neural responses. So here's an example from the famous Churchland et al. 2010 paper on neural variability. And importantly, these transients are not explained or predicted by static inference models. Also, when you plot these transients by contrast level, you see that transient magnitude strongly depends on contrast. The lower contrast case has a much higher transient. So this is a really easily testable prediction of our model that's specific to dynamic inference, and we're working with the Litzberger lab to test it right now. So in conclusion, I presented a spiking model for dynamic inference that replicates behavioral and neural results from smooth pursuit experiments, but also makes novel testable predictions that are unique to dynamic inference. Altogether, our work suggests that it's really important to consider time as an axis over which inference unfolds, and we're really looking forward to testing our model in other contexts. I'd like to thank my advisor, Christina Savin, as well as the entire lab, and our collaborators, Dr. Stephen Litzberger, Timothy Darlington, and Manu Raghavan. Thank you very much.
All right, thanks. We have time for a couple questions. Okay, I have one, which is, do you know in the experiments, what is the time scale over which this prior is established? And also, how does that kind of appear in your model?
Yeah, so we're not explicitly modeling adaptation to the prior. That's a really interesting question, but involves studying learning very specifically. In the Darlington experiments, they did look at adaptation to prior speeds, and that happened over the time scale of about 50 trials to the mean.
It's like we have one.
Yeah, just a quick question. Thanks for the nice talk.
Thank you.
Your work is showing really nicely that there is kind of dynamic inference going on in the visual system, but I'm wondering...
I can't hear you, sorry.
I'm sorry, yes. Your work is showing really nicely that there is dynamical inference going on in the system, but I'm just wondering what it tells us about how it's actually implemented. So do you actually have any evidence that it is, let's say, a particle filter or something else?
Yeah, so that would require more specific experiments that distinguish between the exact algorithmic implementation of dynamic inference, although there are predictions of our model that are very specific to particle filtering that wouldn't be explained, for example, by a simple Kalman filter. And I think that would require, generating more data from more complicated experiments. But that's an interesting question that we want to look at. Thank you.
Okay, well, let's thank all of our speakers again.
Thank you.